Greetings to ladies and gentlemen, our viewers and listeners. Welcome to Bio Korea 2020 live chat. My name is Kyungmi Kim, your MC for today. And I'm so happy to meet you in a very new form today. Well, as the biggest bio convention in Asia, Bio Korea 2020 is delivered online convention from e-conference, virtual exhibition, to business forum, including online meeting, company presentation. Especially this pioneering move, Live Talk, which is attended at 28 companies, provide opportunities to find innovative technologies and products through real-time online streaming. So please search Bio Korea at YouTube for English speaker, and for Korean speaker, 한국어 사용자분들께서는 KBIC를 채널을 검색해서 함께해 주시기 바라겠습니다. 보건산업혁신창업센터 채널에서 한국어 사용자분들 함께해 주시면 감사하겠습니다. And you are invited to real-time live chat. Therefore, please feel free to join us and leave your question or comments to the participating companies. And audience participating our live talk will go in the draw for a gift. So please with us until the end. So today we've got total of 8 companies lined up for today. So it's my pleasure to introduce our first company to present, KBIC을. KBIC을 is developing a tumor position direction sensor that allows us to pinpoint the location of the tumor by attaching a metal cleave to the tumor during laparoscopic surgery. And this presentation will be conducted by Jung Ji-won, CEO of KBIC을. So, hello, Mr. Jung. Good to see you. Thanks for joining us.
Nice to meet you.
Thank you very much.
안녕하세요.
Thank you.
네.
So, please introduce your company.
Nice to.
We will begin now. I am the CEO of KBIC을, Jung Ji-won. Today I would like to talk about my company first. Can you switch to the first slide, please? We were established about 2 months ago. We produced medical instruments, and it is our target. And also we try to utilize the artificial intelligence. And we are developing the early detectors in order to find lesions at the earlier stage. And also we can use the clip to mark the lesions. And these are the goals of our company. And we are familiar with the unmet needs of the endoscopists and surgeons, and we are trying to have the patents and then develop our products. Next slide. And this is the goal and the development motivations. And among the cancers, gastric cancer, such as stomach cancer and colorectal cancer, are also popular, or so in common. And before it was easier for us to detect the lesions. However, nowadays the difficulty to detect the lesions is increasing. So there is a strong need for the endoscopy tattooing. And at the earlier stage, can you hold on a second? There is an audio issue. I'm sorry, the interpretation cannot be provided because of the audio issue. There are several ways for the endoscopy tattooing, and there are several issues. Next slide. So we utilize the big data in order to early detect the lesions, and we develop the algorithm for that. And we are trying to commercialize. This is our goal. And after detecting the lesions, and we will conduct the endoscopy. And we are going to endoscopic tattooing. And then we will have the surgery. That is the process that we are aiming to provide and develop. Next slide. Please, next slide. If we develop technology, we can have the international market with a size of 20 billion. And we can use this device for a lot of population. I believe there is a strong need for this equipment. And 2.5 billion is the domestic market size. And the US market size is going to be about 10 billion. Please move to the next slide. We have the patents. I'm sorry, we have the audio issue right now. The interpretation cannot be provided. And also, we applied and registered the patents in the US, and we already transferred the technology. In addition to that, we have 8 patents, and also regarding medical instruments, and also we have the 2 patents regarding AI. Please move to the next slide. And before we start up the business, we research the prototypes, and we have a full understanding on the detectors and the clips. And the funding needed is about 300 to 400 million, and this is the plan that we are going to illustrate how we are going to fund it. Next slide. And this actually shows the member of our organizations and our capabilities as well. And we have different professors and surgeons from different areas specialized in the gastroenterology. And also we have the engineers for the circuits. So we have all the necessary members in order to develop this device. And also we have research center and medical center, so we can get the device. So we create an environment that is favored to develop all the necessary device that we want. Please, next slide. And we have a lot of strength with our technology. It is relatively very stable, and we can satisfy the unmet needs. And we have a strength. I'm sorry, there is an audio issue. The interpretation cannot be provided right now. From the stage of the designing, we can easily advance into this market. Next slide. This shows the video for the endoscopy. Please play the video now. And this shows the endoscopic tattooing. There is a plug and you inject the ink, and there is an issue regarding using the ink, and you have to use the 4-quadrant injection. However, this is very technically difficult. And the next slide shows the endoclip that we are developing. This is one-touch method. You can put this clip right there. And it is very easy to install, and compared to the traditional method, it is very convenient and it requires a very short period of time. Please, next slide. And I would like to briefly talk about the AI technology. And there are some publishments that we made. And we made up 3 classifications: normal, ulcer, and cancer, and we studied different images. And this shows the accuracy of our AI algorithm. And I think this rate is going to increase to 95% if we study more images. And lastly, I would like to show you how we can utilize AI to detect lesions while doing endoscopy. And did you see the tumor or the lesions? It is very difficult to see this with our bare eyes. However, if we utilize AI, there is a boxes that you can check the lesions or the tumors. Thank you for your listening. If you have questions, please ask us.
Okay. Thank you very much for your presentation. So actually, we're waiting for the questions, but our viewers, I think they're quite shy. So viewers, if you have any questions or comments about the company, just feel free to leave your feedback at real-time live chat on YouTube. So thank you very much, Mr. Zhao, for your wonderful presentation. Thank you.
Thank you.
Okay, so we'll look forward to further investment, further advancement on your company. Thank you.
Thank you.
So, yes. So moving on to the next, we have Hirsarion. And Hirsarion offers a mobile-based healthcare system that is specifically designed to provide physicians and patients with the flexibility and portability. The presentation will be delivered by Mr. Lee Dong-jae, Hirsarion. Well, what I know about your company, Hirsarion, Hirsarion is exporting to 40 nations and showing a clear and rapid growth. And so that's why you are growing as a global company. So can I see Mr. Lee Dong-jae? Hirsarion, hi. Yes. Hey there. Hi, Mr. Lee Dong-jae.
Hello.
Hello. Yes.
Yeah,
We got you here. Thank you very much. So, Lee Dong-jae, would you please begin your presentation?
Okay. Thanks for calling today. We are Hirsarion, manufacturing ultra mobile
질문이 아마 그렇게 많지 않아요.
sound system.
Korea is known to be a developed country.
Next. Next slide, please.
그러면 지금
Yeah, we were founded in 2012. Our company, Hirsarion, specializes in the development and manufacture of portable handheld ultrasound devices. The company's mission is to provide medical professionals with affordable and innovative ultrasound technology that enhance the quality of medical care and improves patients' outcomes in any settings.
이제
Next, please.
하신 거 수고하셨습니다. 네. 영어로
Yeah, our CEO, the Benjamin Jungwon Liu, he has pretty much big career behind him. He started his business as an MD, working in a big national hospital in South Korea, and he founded Hirsarion in 2012. And after foundation in 2012, Hirsarion has won numerous awards for Sunun products, including first place in the 2012 Korea Startup Contest and the Global Venture Company Award. Hirsarion currently holds 64 patents and licenses in
Worldwide,
Korea and worldwide. Next,
therefore,
please.
cancer is the third highest, one after lung, only after lung cancer and
Next, please. Yeah, I would like to introduce very briefly about our technology and products. Next, please. Yeah. Our sonar system, which is based on Wi-Fi portable technology
colon.
in ultrasound scanning. The portable battery-operated sonar is a mobile-based ultrasound device, which is compatible with iOS, Android technology. That means it can detect pneumonia, a key COVID-19 symptoms without an ultrasound or CT scans that are immobile. That means our system can organize and use in many sites, like disaster sites or developing countries, like some in country in Ghana, Gabon, Mozambique, like that. Also in primary healthcare crisis. Next slide, please. Here is our main feature. Hill Science Sonon ultrasound system is really compact and really light comparing with conventional ultrasound machines. You can see the big heavy machines on left side on the slide, but our system on the right side, as you see, our ultrasound system is really compact and light, portable as well. Next slide, please.
Wonderbio's technology is developed
Yeah.
to resolve this.
As our product line can function without an electricity
so
source
that
or
doctors can
internet
cure
or
with existing
hours
technology they have.
of battery charge. It's a really strong solution for patients streaming in remote
Korea is known to be a
and
developed country
non-traditional
and shows
settings,
remarkable achievements in many areas.
private hospital
Incidence rates
admission or at-home screening
stomach
of
rate, stomach cancer,
suspects of COVID-19 infections. It's really small, to fit in your pocket.
That's why we started our business
It's
from
highly affordable and scalable and flexible to meet most imaging needs of a healthcare provider. So we are getting a lot of heaps of inquiries from so many areas, especially in USA and
early
European
detection of
countries.
stomach cancer.
Next slide, please.
Worldwide death toll stomach cancer.
Yeah. The advantage of our product are quite simple. It's like ultra portability, battery operations, high quality wireless connection. That is 4 main features of our products. Next slide, please.
마이크.
Next, please. That's a comparison chart that shows how compact, how light, how proficient our system comparing with players in the market these days. Our system, Hill-Science Sonon, named convex linear type ultrasound system, which has wireless functions and quite portable and light. Also, it has a longer battery operation time than any other players in the market. Also, our price is quite competitive to any other competitor in the market. Next slide, please. We are exporting over 40 countries in the world nowadays. We have 2 overseas branches in UK and US. They are dealing with a lot of things with the stock dealers into all 2 to 4 areas, including Africa market, Middle East market, and Russian market as well. So we got lots of certificates related to quality. We got FDA certificate in 2016 and CE in 2017. Also, CFTA for Chinese market is on the process and might be finalized at the end of this year. Next, please. Next slide, please. Yeah, also, we have lots of patents, which means intellectual property. Next, please. Next, please. Yes. As you see, our Sonon system is the world's first smartphone connected ultrasound medical devices. It was released in 2014. And in 2015, the Sonon 300C ultrasound imaging system became the first portable handheld ultrasound device to be approved by the FDA for clinical use. And since 2017, our market has expanded product capability to include lung scanning, which allowing for the early detection of pneumonia, a key COVID-19 symptom these days. And the production and development of new ultrasound system, which is 30% lighter, 30% smaller, is on the process. Next, please. Next, please. Yeah, that's it. Yeah. We have mainly 2 product line, which shows Sonon 300C and Sonon 300L. Sonon 300C covers the field of examinations and obstetric and general abdomen and trauma scanning field. And Sonon 300L covers MSK market and vascular anesthesia and sports medicine field. So these days is the demand of sports market and well-being market as goes. The Sonon 300L demand is rising at the moment. Next, please. Our main target, it's a little bit of developed country for the last 10 years. But the market is changing every month, so we are focusing a little bit more wider area, including Asian Pacific regions and China market as well. Next, please. We have 2 overseas branches in USA and UK. So overall, 60% of overseas market has been covered by our overseas branches. And the other markets, including Asia and Oceania, including Australia, is covered by our headquarters located in Seoul. Next, please. Also, we are focusing on veterinary market as well. So we signed a contract with a big veterinary company in USA, which is Heska, in 2017. And we are searching for bigger veterinary market in Australia and Europe as well. Next, please. Due to the portability and mobility of our system, much wider market can be made in the next 10 years, which covers general practitioners and pediatrics and pain control clinics and maternal care clinic, also geriatric hospitals as well. Next, please. Next, please. So as the market grows, some major countries on the world like UK, China, next please, next slide please. UK, China, Africa, some markets demands are rising at the moment. And all information and technology should be mixed and united with our AI technology. Most hospital and healthcare system use our system with PACS and EMR technology in hospital. Next, please. And these are our additional market, which doesn't include at the moment, but we are trying to get on this market, especially sports medicine market, transportations and emergency, and public health care clinics all over the world. Next, please. Next, please. Next, please. So, lastly, our next generation of a sonar system would be go with AI. So, I'm pretty sure the next generation of sonar system must have higher image quality with better usability, with lighter weight and smaller body, as you see on the screen. Next, please. With AI technology, most doctors in developing country can learn and control ultrasound system very easily. Because AI guide procedure, it's much easier than current ones in the market, and they can learn and communicate with so many professionals and clinicians in the world. They can upload their clinical data through cloud database, and they can also download lots of clinical cases from the cloud, which can help the standard of healthcare system in their market or countries. This is end of our presentation for sonar ultrasound system. Thank you for time.
Thank you. So, Mr. Lee Dong-jae, thank you very much for your presentation. So you are indeed expanding this market as a pioneer, and your device indeed has a lot of competitiveness. So I look forward to seeing your expansion. Mr. Lee Dong-jae, thank you for your presentation. So now I am checking our YouTube channel. So we have many viewers here. Thank you very much for your comments. But it's not a serious comment, just, simple words. Hi, Jinchul. We have a Jinchul here, Dong-jae. Oh, our speaker here. And Hanna. Well, we call Ipsi Medic. And hi, Chang-ho. Hi, Winnie. Thank you very much for your participation. So any comments and any questions are all welcome. So just don't be shy and leave your messages. Again, we have 2 channels. So this live talk is being conducted as a real-time streaming service. So please search BioKorea at YouTube for English speaker. And for Korean, 한국어 사용자분들께서는 KBIC 보건산업혁신창업센터 채널 검색하셔서 들어가시면 한국어로 지금 이 방송 함께 하실 수 있습니다. Thank you very much once again. So, what's your next step? Yeah, Mr. Lee Dong-jae, can you just answer that question? Your pipeline's future plan. What's your next step of this area?
We are trying to expand our market
Mm-hmm.
in areas
Mm-hmm.
like the market.
Mm-hmm.
And yeah, we are kind of pioneer in the market.
Mm-hmm.
So we are focused on new markets, like
Mm-hmm.
sports market.
Okay, so I look forward to seeing your expansion to the global market. Okay. So it is time to say goodbye, Mr. Lee Dong-jae over here, Saryon. Thank you very much once again.
Thank you.
Yeah. Thank you. Okay, so now the next company we're going to meet is Handok Medical. Well, Handok Medical is one of the medical device manufacturing company which mainly produce products used in hospital. So this presentation will be covered by Mr. Shin Ju-pyong at Handok. 자, 한독 만나보도록 하겠습니다.
Good afternoon. Oh, your name is Ipyeong. I'm sorry. So, Ipyeong 님.
I miss Khalid.
Yeah, Lee Pyung-hwan, thank you very much for joining us. So, would you please introduce your company and your technology? 네, 발표 시작. 네.
You can start right now. We have been operating in this business for about 37 to 38 years. Can we continue? Shall we go on? Can you pass to the next slide? Next. This is the list of the products that we are producing. First, a ceiling pendant system. This is the only system produced in Korea and by the Korean company. Compared to other competitors or other product, it is differentiated with a unique technology. In particular, I would like to stress on what is differentiated. It can suck up the unnecessary smells, and also it can be used in the surgery room, and it can remove all the gases, and also it could prevent from collision to the walls by using the ultrasound. This is a special functions combined in the system. Also, we have bacteria and virus filters for the respirations or anesthesia. And also it can be used PFT. And it can be used in the emergency rooms. So we are producing a variety of filters for the different purposes. Also for the endoscopy, we are producing the mouthpiece or other devices needed in order to provide the oxygen. So for the ventilation, we can provide the oxygen and saliva is automatically removed. And also we can measure the concentration, so we can monitor actually the concentration level. These are the unique technologies that we are developing and utilizing, and this is combined in the endo mouthpiece as well. For the ventilators and and also we are producing filter mask with a tube. It prevents bacteria or the viruses for the inhalation. And we provide the oxygen at the same time without any bacteria or viruses. And also we can prevent the removal of the impurities from the patient's body. And you can categorize our project into 4. Can you pass on to the next slide, please? Next slide, please. And this shows the variety of the ceiling pendant systems that we produce. You can add monitors or the arms to the system. And on the right side, the one can be used in the surgical rooms. Mostly it is used in the emergency rooms, op rooms, recovery rooms, delivery rooms, or the wards. Next. This shows one of the characteristics. Well, actually, we can use this in the operation room. And you can add 2 monitors to the systems. And you can add the shelves, and you can move up and down. And also, it can lift up to 400 kilograms. And this picture shows the actual strength test of our system. So it shows how strong it is, and compared to other products of our competitors, it has a high strength. And the structure as well. We have a patent for the structure of the system. And also we have the bearing systems. It is 2 bearing system actually. So it can be operated for a long times without any mechanical changes. Next. Well, we have only 20 minutes, so can you move our slides faster a little bit? We have this characteristic actually. This is the only functions that we have. During the surgery or operation, we miniaturized the system, and this is what it shows. And also we have the evacuation system with electricity. And all the unnecessary hydrocarbon can be removed, and this is not observed by any of the surgeons during the surgery. So these are the differentiated technologies that we have, and we only have this technology in the world. So we are the only company with this technology. Basically, the ceiling pendant, computer, leg, EMR, PACS, all of this data solution can be included in the systems, and the pump and other functions can be added to the existing system as well. Next. Well, I think there is a technical issue, so and this shows the structure of the bearing that I explained previously. We have a 2-line bearing systems. So compared to other competitors or a single-line bearing, the design itself as well as the functions are so differentiated. So we can have guaranteed years of at least 20 years. Next. And this mechanical structure is the unique technology that we developed and we designed. Also, we have the Korean patents regarding this technology as well. And this diagram shows that we are producing products and we have with the rail systems, and it can motorize up and down to 400 kilograms. It means that it lifts things up and it can move up and down to up to 400 kilograms. This is one of the differentiations that we made to our system. And this is high performing and this is very strong. And this is very economic. In addition, we apply patent and we register technology regarding this next. Next, now this diagram shows that our patent. We have instrument table consisting of 3 plates, and this can be reorganized, and this can be added to our existing Panda systems. So this technology is differentiated as well. In the surgery or environment, we can use this system, and it creates the optimized surgical plates for the surgeons. This is a list of the filters that we are producing. Among them, for the ventilations and for the anesthesia, we develop and produce bacteria and virus filter. And also, these can become with and without the function of the humidifications. And we also produce filters for the PFT. In particular, KR101 is designed in consideration of the actual structure of the teeth of human. And also, the test result of these filters in terms of the BFE and the BFE, it's more than 99.9%. Next. In particular, bacteria and the virus filters are very useful in order to fight against the immunicable diseases such as coronavirus, or to prevent spread of the virus. Next. This shows the characteristic of our products. On the left side is a PFT filter. It can prevent the gases emitting from at a high speed. So it has excellent functions of the clarifications. And the one on the right side was designed in consideration of the actual teeth structure. and also COP2. And we are producing a lot of filters can be used for the ventilator, anesthesia, COPD, ambient bag, and next. This product shows the functions of stopping the coronavirus from spreading out. It has a characteristic of, well, actually, if you compare this to the conventional traditional product, it prevents spread of the viruses in the process of the inspirations. And the saliva and the impurities or the germs can be produced in the process of the expiration. With this device, it can be prevented. So for the inspiration and expiration, it can purify the germs in order to make the medical environment cleaner. It can purify the environment, and this can be used in the emergency rooms in order to prevent the movement of the foreign substances. So these can be used, and this is very special in the era of the coronaviruses. Well, this can be used in the ambu bags, and this can be used for the CPR as well. And of course, this can be used in the operation room and other words needed for the general anesthesia. Also, we applied and registered patents regarding the design of this product. And also, when we go to overseas to participate in the exhibition, Europe and the Middle East and Southeast Asian companies are so interested in this product. I think this will play a key role in order to fight against the coronavirus. These can be seen as a preventive device next. Next. This is what we are producing as well. This is 840 ventilator filters, and when you take a look at the BFE and the VFP, it's more than 99.99%. This can be used for the ventilators. Currently, it boasts the high performing among the other filters that are produced across the globe. When you take a look at the test results, as you can see, we got the result of 99.999% for BFE and the BFE. Next. This shows the special features of the mouthpiece. Rather than its a simple purpose, it has a variety of functions. First of all, during the endoscopy or during the conscious sedation endoscope, you can supply oxygen directly to the mouth, and also it can automatically remove or drain saliva, so it prevents any leakage of the saliva from the patients. So there is a tube inside it, sucking the saliva to prevent leaking of saliva. So it is very hygienic. Also, it monitors carbon dioxide produced during the endoscopy, and from the patient monitoring, they can monitor the level. So we can tell the patient is breathing or not. And also it has a characteristic of that it is very easy to fix in the mouth of the patients. So without any hairbands. This is not really removable during the endoscopy. And we have a patent in Korea as well. Next. And as previously explained, this is the system that you can use with the endo mouthpiece. It's what you can control the endo mouthpiece. It is safe and it is economic and it has a lot of functions. It can control the level of oxygen, and it can remove the saliva with a tube, and also it can remove impurities or the saliva and collect them in a bottle. Next. This diagram shows the auto controls device and mouthpiece can be used during the endoscopy. And there is a connection tube. This comes in one set. All of this, once again, can be used in the endoscopy. As far as I know, we are the first one to develop these set and device. So we can supply oxygen, and we can remove saliva automatically at the same time, and in the emergency cases, it can have a function of sucking, and the saliva can be collected in a bottle, as you can see from this diagram. And it is very economical. You don't need any nasal tube during the conscious sedation endoscope. So it streamlines the process and it is really safe for the patients, and for the surgeons it is very convenient, it is very easy to use, and this is all economical. These are the advantages of this system. Next. And this shows the control device for the endoscopy, and also this includes the connectors, and one time we have a mask for the one-time uses. And after installing this device, most of the functions can be done automatically, so it reduces time or the efforts needed of the surgeons or the physicians. Next. And this shows the details of the functions. It can control the level of oxygen, and you can monitor with the flow meters on the device. And also there is a switching or knobs for sucking saliva, and also there's a flow meter to monitor this level as well. And there is a switch for the emergency purposes. And the impurities, the germs can be collected in a bottle, which is collected automatically. And this is disposable, so we don't have to pollute the air of the room. So we can prevent the spread of the viruses or the germs. So this is very special and needed function in the era of the coronavirus. So we can prevent spreading of the virus actually. Next. If there are foreign substances from the carriers or the patients, well, we can use mouthpiece and the reusable connectors and the bottles. All of these are disposable in order to prevent spreads of the germs of the carriers or the patients. So this is a very basic device to prevent that. Next. And that was the end of our presentation. Thank you.
It's working on hypertension treatment device in Korea. I think the company is looking forward to tomorrow more than today. 네, 감사합니다. 이평환 대표님.
Thank you. It was a presentation of the CEO. Can you hear me?
예, 이제 한동 메디컬 나가도 됩니까?
네,
Can we go out?
about your presentation. 발표 질문이 굉장히 많이 올라왔습니다. 그래서 와, 지금 한 7, 8개 정도가 올라왔는데요. 제가
We have like 7 to 8 questions, but I will took 1 or 2.
viewers, we have there are many viewers getting more and more. 자, 레귤러 필터 체인지 사이클에 대해서 물어보셨습니다. 민희님. 네, 그 필터를 교환하는 그 주기에 대해서 여쭤본 게 맞으시죠? 사이클. 대표님, 대답해 주시겠습니까?
필터 종류는 원천적으로는
네.
일회용입니다. 싱글 유즈 디스포zal.
This is disposable actually. It's for the one-time use. And we got the excellent feedbacks about the filters and the mask.
Able to children and adults? Is there any size for the mask or only one size to use all? 네. 질문해 주셨습니다.
Miss Cho asked me about the size of the mask. Does it come in one size currently? We have only one size. And there are so many people who are interested in the Hando Medical. So how should we contact? Well, actually, if you participate in the business line of the Bio 2020, you can contact Hando Medicals. If you're well, I believe there are a lot of people interested in the Hando Medical. So how can we contact it? Should we go Google your contact information? I think the email will be the best option. And we listed our email in the website of our company.
You can contact the company. 자, 바로 이렇게 검색하셔서 들어올 수도.
Yes, you can Google our company.
2020, and then you will have chance to connect the company participating live tech. Okay. 네.
그래서
Can we tell them our email address right now? We can connect this person to you, but if you have someone in charge, we can ask that person. But which way do you prefer? We can tell email right now. So can you leave your email on the chat room?
At Hando Medical directly. 네, 지금 여러분 보시다시피 대표님께서 직접 이메일을
Yes, the CEO is writing down the email to the live chat room. So I think you can use the email to contact the Hando Medicals.
Hong 그리고 윤하조, 민휘강, 민휘강. 자, 많은 분들께서 코멘트를 주셨습니다. Once again, thank you very much for your active participation. 자, 대표님 이렇게 해서 이 시간 마무리하도록.
So that is the end of the presentation of the Handong Medical.
Thank you very much. 고맙습니다. 열심히 작업하고 계시는데. Bye. 고맙습니다. 자, 이제 MS 메디로 넘어가면 될까요? 자.
So shall we move on to MS Medi now? Okay, so goodbye to Handong Medical for now. So let's move on to the next company.
And Mr. Han Moon-seok is now standby for the presentation. Hi, Mr. Han. Mr. Han? 자, 지금 입장을 하고 계십니다. Hello, MS Maddie. 들어오셨나요? Are you on? 자, 바로 발표를 시작.
We're gonna start right away.
Is on the way to get in the channel. 자, 다음 MS 메디 한문석 님께서 지금 입장을 준비를 하고 계신 것 같습니다. 다시 한번 채널
Once again, we're still waiting for Mr. Han for his presentation.
as a real-time streaming service. So please search Bio Korea at YouTube for English speaker. 자, 그리고 한국어 사용자분
And for Korean speakers, please search KBIC.
we have two channels for the English speaker and for the Korean speaker, Korean people. And also, you can leave any comments and questions about the company, about the presentation. And also, we prepare some gifts for the active participants. So it's now a little bit delayed because it's kind of new form and new try. So we're now checking technical issues. So please wait a moment. 네, 저희가 지금 실시간으로 프로그램
Once again, we are streaming real time. So please understand that there's a little bit of a time delay here.
for in the Bio Korea 2020 Live Talk. And we have 28 companies providing presentation for the next 3 days. And we have 8 companies waiting for you today. So before moving on to the next, let's watch some promotion video. 자, 저희가 광고 영상 먼저 보시고 다음 발표 이어가도록 하겠습니다. Please watch the video. So now we're coming back. Thank you very much for your patience. Okay, so next company we're going to meet is ASCO. ASCO supplies hospital negative pressure machine in the event of MERS in 2015 and COVID-19 in 2020. I think it's kind of hot spice company. National Medical Center, Korea Centers for Disease Control and Prevention, public health centers, military base, medical institution, hospitals, and local government. So I'm quite curious about this company. So now, Cristiano. Hi there. Hi. So, can you introduce your company, ASCO, please? Okay, Cristiano, hi. Can you just turn on the microphone? We cannot really hear you. Can you just please turn on the microphone? Hi, Cristiano.
Can you hear me?
Yes, now. So can you just start from the first?
Okay, sorry, thank you.
Okay.
So we at Asmedi are specialized in manufacturing and distributing medical equipment and consumable supplies for infection control for medical facilities and biological laboratories. Our company has supplied more than 2,000 negative pressure machines to national health centers, public health centers, and other 300 medical institutions and hospitals nationwide, especially during MERS outbreak and COVID-19 outbreak. During the MERS outbreak, we already had about 65% of the market share when it comes to negative air pressure machines. And now in COVID-19, we reached 92% of the market share. That is the reason why our main goal is to introduce our products globally in relation to our successful knowledge of this particular field. From now on, I will go on explaining shortly about the advantages of using our products. The first part would be that, for example, an isolation chamber would cost around $250,000, while one of our machines alone can deal with $2,500 to $3,000, which means it's a very cost-efficient way to create an isolation chamber. Second one would be that we have to keep in consideration, for example, the medical staff. The medical staff is very important because if they get contaminated by the virus, they would be very, they would create a lot of problems, for example, even during the treating of the patients. This is very important. Even the airports and ports are currently an issue because our machines are installed in all these places. And in Korea, they blocked the spreading of the virus, but in other countries, would be a little bit of a different situation. It is important to think about, for example, the cost of the medical staff. Every medical staff in every hospital costs around $45,000 a year. This is an important cost, and creating an isolation chamber would be very important for this matter. So basically, our machines are currently being used in the major hospitals. And thanks to them, the contamination in Korea has been kept very under control. And before this Itaewon situation came out for the recent outbreak, we basically helped the correct handling of the situation because our machines, as I previously mentioned, are in 92% of the hospitals in Korea. So we are currently trying to expand our global network because our global network is relatively small for the moment. But we are very confident that if we could find some reliable distributors, these machines would be very, very used in all these hospitals. Of course, due to that, even an eventual distributor should get in contact with them. We have several advantages to our machines. And from now on, I will ask the help of one of my colleagues to show you the installation and the functioning of these machines. So from now on, I will show you a little bit how they work, and they can get a glimpse even now during the live. So this is Roberto, my colleague.
Hi everybody, my name is Robert. Very nice to meet you. And for now, I'm not going to introduce our products. As my friend mentioned, we have two products. The first one, you can see this is the 2501, and the other one there is the 1502. We're going to start with the installation process. So the first thing that we have to do is to move the machine in the place where you're going to install the negative pressure machine, and then you have to take it off. You have to take off the cover, and also you have to take off the bolts. So you can see here the bolts. And after that, once it's reinstalled, then you have to install the hose. You have to make sure it's properly installed, and also bring the hose here to the window side. You can see it there, the window side, and make sure that it is properly sealed and is fastened to the hood cap. Once it is properly installed, we're going to look at the operation process. So the first thing you have to do after the installation, you have to make sure that this wire is properly installed here in this section. And then there is a switch on to turn on and off. When you turn on the switch, you come in front of the panel, and then you turn on the main system switch. You turn it on, as you can see, it's already in red. And then you press the power button. You can see some numbers there. And after that, you press the second button, and it will automatically appear from 100 and 0. So we have to wait around 10 seconds so the system can start operating. We're gonna wait around 10 seconds, and then you will see how it will start operating. The outflow is going to go up. So now it already started. You see, probably you can see 5, 7, and then we'll go to 10, 15, and up. The more it goes up, the sound increases. So now we have to regulate with the up and down bottom. You can regulate here, 80, 75. And you can also leave it to 5, but it is recommended to leave it from 25 to 35. So now we're going, just a sample, we're going to put in 25. So yeah, that's how it operates. This is the 1501 operation process. And now I'm going to show you the operation process of the 1502. So the process is very similar to the previous one. What we do is to connect this wire. It's already connected, but we connect the wire, and then we turn on the switch, and then we also turn on the main system. Once it's turned on, we press the power button and the start and stop button. We have to wait around 10 seconds. So you see, now it already started operating. It's 5, 7. It's the same process. It is basically the same operation process. So you have to regulate it from here, the up and down button. So now we can go down to 90. And the more it increases, the more the sound it is. But you can also regulate. You can regulate it to 25, for example. So we're gonna put it in 25. You see, you're regulating the sound. One very important thing I'm gonna show you is that when you install the hose, this machine has two sides, the inside and the outside. So the hose has to be installed from this side just to make sure of it. And the hose has to be connected here and connected to the hookup to the next to the window, okay? So for now on, I'm going to explain about the filter replacement process, which is very easy, no technical skills are required. So you open the front door. There are 3 levers here. You open all of them. And before that, you also have to make sure that this bottom is turned off because we're going to change the filter. There's no major problem, but you just have to make sure. So you turn it off, and then you open the front door and open. You open the front door, and here we have, as you can see, we have 3 filters. The first one is the pre-filter, medium filter, and the HEPA filter. The life cycle of this filter is 1 month when operating 24 hours. And the medium filter and HEPA filter, you can use them for 32,000 hours, which means it's about 4.5 months. And the other thing is how to take it off. So you see down there, there are some levers. We're gonna make sure you pull it off, just like this. And now you can take off all the filters. This is the medium and the HEPA filter. So one thing you have to make sure is, if you can see the blue arrows here, when installing the filter, just make sure these are towards the main motor, because if you change it, probably the capacity, it wouldn't be so efficient as it should be. So you just have to make sure. And the other thing is, these filters here, they have a ceiling here, so the air does not leak out. There's no way the air is gonna leak out. So the operation, you also hear the efficiency of the HEPA filter is 99.97. So once it's installed properly, you pull it back and you proceed to close the front door. And that's it. That's basically the operation process of our 2 machines. And thank you for listening. And if you have any questions, you can proceed now. And yeah, my friend is gonna talk for now.
Yeah.
Thank you.
Okay, so as my colleague has explained so far, it is also important to know why these machines are important. Since they create negative pressure, and in some cases they can also create positive pressure, like the other one on my left. Negative pressure is important because it creates a vacuum in a hospital room. So every virus or contaminant that is present at that moment can be practically sucked out. In order for negative pressure to happen, we need to have 30 to 40% more airflow coming out of the room rather than the airflow coming in. So we create the vacuum effect. The positive pressure basically works in the opposite way. So there's more air coming in, and it's mostly used in infantry rooms, so for very young people, or for
그럼 MS 메디 할게요. I'm sorry.
because we have to keep them safe, and we have to make sure that they are, they are the people who are most affected by these kind of situations. So in order to make sure that possible distributors get familiar with our products, we are currently having a promotional period. And in this promotional period, we want to offer both of the machines at half the price as a sample order, we would say, so they can get familiar with the functioning that we already showed a little bit in this video. And if they consider it proper for a bulk order, we also want to keep a good relationship with any possible distributor by offering 1 free machine on the top of the warranty that every machine has, every 50 machines that are purchased. So it means that if a machine had a problem of any type, there could be a replacement ready practically on the spot. And yeah, due to this, we are offering to also keep in touch. And if there is any question, we would like to reply to them. They can also write both to me or my colleague. My mail would be Cristiano, Cristiano, at ASCO, ASKO, 119.com. For my colleague, it's the same mail address, but it will be Roberto, so ROBERTO, at ASCO119.com. That is practically everything we wanted to show you today. Thank you for listening.
Thank you very much. We have listened about the details of ASCO's core technology. So, Cristiano, could you please leave your the address that you just mentioned on our live
sure.
chat talk? Yeah, so that our viewers can contact you directly, right?
Sure. The address mail will be CR.
Can you just leave the address on our live chat?
Oh, okay, sure. We will write it down.
Okay, thank you very much. I'd like to thank our two gentlemen for the kind introduction. And we have many viewers here. Thank you very much once again. Well, Mark mentioned that these are equipments to protect the medical staff and non-infected people, which keeps the air pressure in the patient isolated room negative, so that the contaminated air in the room does not leave the room. Yeah. Can you just read it? So we have messages.
Oh, yeah. So basically negative pressure works in that way.
Mm-hmm.
It's a way to keep the contamination room completely isolated from contaminants. And what was the question exactly about?
So these are equipments to protect. It's not, I think it's not a question, but just comments. These are equipments to protect the medical staff and non-infected people, which keeps the air pressure in the patient's isolated room negative, so that the contaminated air in the room does not leave the room.
Exactly.
Yeah. Yeah, that's okay. Mark that. This is something that you understood, correct?
Yeah.
Thank you. So Yoon-ha Jo also said that address one more time, please. So please leave the address on our live chat. Okay?
Oh, okay, we will.
Okay. Thank you very much for your time. Bye.
Thank you. Very much.
Thank you.
I will leave the comments.
Okay. So, ladies and gentlemen, let's move on to the next company. The next company is Wonder Bio. Wonder Bio developed diagnostic solution to detect cancer only. Diagnostic machinery or system to detect cancer only exists. So now, Mr. Lee Young-gyo here with us for the presentation. Hello, Mr. Lee.
Hi.
Hello, can you hear me well?
Yeah, very well.
Okay, please begin your presentation. 발표 바로 시작해 주십시오.
Okay.
Please start the presentation now.
Our test device can detect cancer earlier than any others, easier to use than any others, and quicker to see test results than any others. We can provide this test as low as $20 to $30 per test. At such a low test expense, we believe everyone in any countries can use it. Next, please. $9.9 million died in 2018, and 11.4 million are expected to die because of cancer in 2030. COVID-19 killed 32,100 people worldwide until now. This number is only 0.3% of cancer victims in 2018. Wonder Bio's technology is developed to resolve this global issue by detecting cancer at the earliest stage possible so that doctors can cure cancers with existing technology. Next, please. Korea, the slide doesn't change from the beginning. Okay, next. Okay. Korea is known to be a developed country and shows remarkable achievements in many areas. Incidence rate of stomach cancer is one of them, unfortunately. That's why we started our business from early detection of stomach cancer. Next, please. When normal healthy cell turns into cancer cell, it produces cancer-specific gases. This gas comes out of the cancer cell and goes into the blood vessel. The gas comes out of the blood vessel when they arrive at lung. It is critical to detect cancers as early as possible by using these cancer gases in order to cure cancer as early as possible. Next, please. After 23 years of R&D work, our scientists completed in last year development of biosensor that can detect cancer-specific gas from breath. The prototype of breath analyzer is expected to be finished by the end of this year. Next slide, please. We need to do clinical trials with breath test device from 2021 for 1 or 2 years. We expect sales license will be obtained by the end of 2022 for sales. After stomach cancer, we plan to develop biosensors for lung cancer, colon cancer, and other most common cancers in Korea and the world. Next, please. Our sensor is a copy of human sensor, which is more powerful than any man-made sensors. Human has 390 smelling sensors called olfactory receptors in the nose. These 390 sensors can detect or recognize 1 trillion smelling stimuli. We see how powerful human smelling sensor is. This disk type in the right side of the slide is the most advanced type of biosensor. This type can smell at lower concentrations than any other sensors. We have patented technology that we can produce biosensor in the most efficient and economical way. We clone human olfactory receptor and fabricate this disk type sensor in E. coli so that nobody can beat us in pricing. That is why we can offer breath test for cancers only at $20 to $30. Next, please. Dr. H. Joe is a medical doctor and PhD in medicine. He was a professor in the University Beck Hospital. He has been working as president or chairman of a number of academic associations. This strong network with professors and medical doctors will help market breath test device of Wonder Bio. All these 3 people are co-founder of Wonder Bio, and they work for our company. Professor T.H. Park is professor in Seoul National University in the College of Engineering. He is the inventor and patent owner of Wonder Bio's biosensor. He developed biosensor for stomach cancer with Dr. H.K. Young. Dr. H.K. Young is professor and director of Stomach Cancer Center of Seoul National University Hospital. He developed biosensor for stomach cancer with Professor Park in Seoul National University, who is co-founder of our company. He will serve as chairman of Korean Academy of Cancer for the next 2 years. As he is well-known medical doctor in Korea and abroad, he will be of great help for WonderBio in developing and marketing blood test device domestically and internationally. Next, please. As we have developed the most important part of blood test device for stomach cancer, we'd like to ask for $2.5 million from investors around the world. If you have any questions, let me answer you.
So are you done with your presentation, Lee?
Yes?
Okay, thank you very much. So we have many viewers here. Viewers are paying attention to WonderBio. And I look forward to your future. So do you have anything to say to our viewers or anything to emphasize in the end? 마지막으로 하고 싶은 말씀 있으시면은
If you have anything to emphasize, please use this time.
Only a startup began in last year,
Mhm.
but our product or technology is based on the continuous and consistent R&D efforts by our co-founders for more than 23 years.
Mhm.
That's why we have developed biosensor.
Mhm.
So we have a newborn baby. Yeah, kind of a middle school student, and brilliant, and expertise we have, but we don't have device. The
Mhm.
device is only housing of sensing sensors, so that we don't have too much difficulty in developing that and complete it by the end of this. That's what I want to exaggerate.
Okay, thank you very much.
The next
All right.
slide,
Mhm.
you will see my contact number and address
Mhm.
and everything. So if you're interested in investing or asking me questions,
Mhm.
please contact me by the email.
Okay, so did you can you just leave your email address on our live chat or
Yeah. Well, you can
Okay.
see at the last part of my presentation, last slide.
Mhm.
I wrote it down, the emails and everything.
Oh, okay. Okay. Thank you very much. You humbly said that it's a small company, but I think it's not small, but it's really, really strong one. Thank you very much, Mr. Lee, for your kind introduction. WonderBio. Thank you.
Thank you very much.
Okay. Viewers, moving on to the next. We have 6 companies waiting for you, Mr. Han Moon-seok from MS Medi. So MS Medi,
네, 들리나요?
yes, I listen to you very well. I can hear you. So Mr. Han, so since I don't have detailed information about your company, so what is the main product, main technology of your company?
In English or Korean?
Oh, okay. So either one is fine. So did you prepare the presentation in Korean or English?
Korean.
Okay. Then just something that you feel comfortable. 편하신 말씀으로 해주시면 되겠습니다.
네, 그러면
Okay, so let me start my presentation.
대전 발표를 시작하시겠습니까?
네,
Okay, so let's.
Good afternoon, everyone. We are a company that is constantly working on developing new form of medical devices. And that is our focus of the company. And my name is Han Moon-seok. And what I'd like to explain to you today is the fascia closer built-in trocar, and the brand name is MS Crocker, and the Crocker is a combination of closer and trocar, and it embodies our main technology. Next, please. Okay, so basically the MS Crocker, it is a device that is used in open surgery. So let me just explain how the surgery is performed. So conventionally, if you think about the open surgery, I think you know about this pretty well, but the incision area is quite big, and if there's a lot of bleeding and bigger scar, and that would also increase the time for the operation, and that has been some of the challenges. So we thought about the new way, and as this new form of the surgery has been carried out, unlike the open surgery, the wrapero is done differently. It is done by making a couple of incision points, and then the medical doctor would include the camera and other medical devices, and that's why it is called the minimally operation. And you can see how the robot is used in the operation room, and this robot use can also be classified as one way to perform the wrapero surgery. And comparing to the open surgery, smaller incision and smaller scar and less bleeding and shorter wound healing time, as well as the less pain, those are the advantages that this new methodology, the wrapero, is being used. Then to perform the wrapero surgery, we need to have trocar, and it is basically placed through the abdomen during the surgery so that it can actually make a portal for other medical instruments. And you can see this picture here. And after the operation and after removing the trocar, you have to close the fascia. And what fascia is, it is basically consists of the tissues, and it is a layer that comes below the skin, and you have to close that area. And as I've mentioned in the very beginning, trocar, or the incision area, it is only like a centimeter long, so it is very small, so that being in there and closing the fascia, it is indeed a complicated procedure. So quite recently, this new way and the need for having this new way to perform this has been discussed a lot. And so I've been thinking about these challenges, and to overcome these, what I did was I developed the MS trocar. So because this suture is already equipped in the trocar, and when it's caught, then after removing the trocar, what happens is you can simply use the function of the suture. Then that way the closing can be done quite easily. And please, let's move on to the next slide. Can you please move to the next slide? So please move to the advantages of the product slide. Next one, please. Next. 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 Yep, that's it. So once again, to sum up, the MS trocar, it does have the unique function of trocar, of course, which can make a portal for the subsequent placement of other instruments. And again, with this one-touch use, the closing of fascia is possible. And next, please. Next. Please move on to the next slide. So for now, if you think about the competition and the landscape in general, you can actually divide the market into three. So the mostly used trocar market is one, and non-built-in trocar. And what I meant by non-built-in is that there's trocar, just like our product, but after removing the trocar, there has to be the separate use of independent device, and that's what I meant by non-built-in. And another one is the built-in trocar, just like our product. And there's a competition called Tradin in Israel, but they actually have some disadvantages in their products. And as you can see, the standard trocar, it performs the unique function of trocar, which is basically to make the portal. And if you look at the separated fascia closer device, it does not do the unique function, but it only can close the fascia. But if you look at the fascia closer built-in trocar, not only the unique function of trocar, but also it can also function of closing the fascia. But if you look at this Israeli company, it uses absorbable anchor. And that's why it is difficult to penetrate into fascia using their product. And the absorbable period is pretty long as well. But our product, we are using already proved suture so that we can overcome these challenges that our competition has. And also, if you look at the market size, domestic market size of the surgical instruments for Raparo, the total size is around 120 billion, and the global market, the total size is around $15 billion, according to our estimation. And please, next slide. So this is our development plan. By the end of 2021, we're planning to complete the approval procedure, and through active marketing, we'll be preparing the basis for the commercialization. And starting 2022 to 2023, we'll be launching a product in the Korean market, and afterwards, our plan is to expand into the global market. Next, please. And this is the sales forecast that we have in our mind. And to put it briefly, by 2022, our goal is to sell products around 22,000 units. And this is the sales revenue that we are planning. And next is about intellectual property. For now, we do have one registered patent and related patent. There are three that are currently in process. Our first goal, other than this, will be the US market, and then the Chinese market, and Japan in regarding to applying for the intellectual property. And well, to talk about my background briefly, for the past 25 years, I've been spending my career in the medical device market, and I have done numerous market feasibilities, and I have indeed accumulated various widespread market experience. And there have been some products that I have launched firstly in the Korean market. For example, the ultrasonic bone cutter, ultrasonic navigation, cryosurgery, 3D endoscope, and Pactus excavating device, and non-invasive surgical skin closure. This is my experience regarding this industry. So, and because of this, I always had this passion for developing new form of medical device. And if you think about the visibility or viability of the product, first you can see that we are indeed registered as a venture company in Korea, and we are registered here and there, and our patent is registered as mentioned, and with the Dongguk Hospital, and with this startup health center, we've been getting professional advice from the medical professionals. And next, please. Next. And the next is, these are the two products that we have actually completed registration and approval by the FDA. And the first is the drainage tube fixation and surgical skin closer. Basically, it is to facilitate the fluid removal. So basically now how it is used is you basically fix a drainage tube by suture, and after the tube removes, then the tube side is closed by the suture staple. But according to this process, there is a high risk of infection, and it takes usually long to recover. So these have been some of the challenges on the site. So what I did was maybe after fixing and after removing the drainage, these two functions included drainage tube. This is what I came up with and I've made, and it's already registered, and also it is registered in China as well. And one advantage is that there's lower risk of infection, and the procedure time is shorter, and there's less pain for patients. And another thing is the surgical skin closer. Now, either the suture or staple is being used for closing process, but those two, there are some disadvantages, including the high risk of infection, pain, and the cosmesis is not very good. So what I did was that I've developed this non-invasive surgical skin, and one big advantage is that the speed of the procedure can go up, and also from the cosmesis perspective, it is better than these two ways, and there's much lower risk of infection, and there's no pain when removal. So we have overcome those disadvantages that are often experienced with the current major ways. And this is the end of my presentation. Thank you for your attention. Thank you.
Very much, Mr. Han, for your kind introduction. Well, it seems that you have already acquired a well-acquired technology, which is more than just putting your efforts. So this is the company MS Medi, with a lot of know-hows, and indeed we'll look forward to seeing new innovations from you. 네, 발표 너무나 잘 들었습니다. 자, 질문은 없고요. 좋은 말.
There's no question, but I think there's this comment that is worth sharing. So the comment says that I'm hoping that you will see a huge growth going forward. And there's another comment that goes, good. So any final comments?
Lastly.
네, 아직
Well, for now, our company is relatively small. So clinically or technically, we are actively looking for overseas partners. So we're not just talking about domestic partners here. So if you have some interest in our products and our technology, please feel free to contact us.
We're a very good partner, not only in Korea, but also worldwide. So if you are interested, you have a very good way to contact Mr. Han. Please register our business forum at BioKorea 2020, and you will still have a chance to connect companies with core technology, high potential, like MS Medi. 네. Once again, thank you very much, Mr. Han, for your presentation. 네, 더 큰 발전이 있길 바라겠습니다. 고맙습니다. 네.
Thank you.
Okay, thank you very much. 자, 이제 벌써 7번째 회사가 되겠습니다. So
We have the seventh company ready.
today, so our seventh speaker is Eric Haliowa from PDC Line at Belgium. And PDC Line Pharma is a clinical stage spin-off of the French blood bank that develops a new class of potent and off-the-shelf therapeutic cancer vaccines based on proprietary cell line of PDC Line. So, Eric, hi there.
Hi, hello.
Hello,
Good morning.
Good morning
Can you
there.
hear me?
Yes,
Good morning.
I hear you. Well, here in Korea, good afternoon. Hi, thank you
Good
for
afternoon,
joining us.
Yeah, it's
Okay.
morning.
Yeah.
Hello from Belgium.
Oh, yeah, you're from Belgium. Well, good to
Yes.
see you here. Thank you very much. So could you please start your presentation? We have, just for your information, we have kind of enough time so you can actually make your presentation longer than you prepare. So just feel free to do your presentation. Okay?
Okay, thank you very much.
Thank
So
you.
if we can go to the next slide, please. I will present PDC Line Pharma in a snapshot. So the company is a spin-off from the French Blood Bank. We are today a team of 24 people, including seasoned executives from biotech and pharma companies. I will present the management team a bit later. In terms of funding, we get close to 32 million euros of financing, including equity and non-dilutive money. And we should mention that the last round of financing, the round B we closed end of last year, was led by Korea Investment Partners, one of your big VC venture capitalists in Korea. So the company has been developed based on the discovery of the scientific founder in the blood of leukemia patients. He discovered, and he was the first to characterize an extremely rare leukemia called plasmacytoid dendritic cell leukemia. You know, in the blood, you have different kind of dendritic cells that are presenting the antigens to the T lymphocyte. You have the plasmacytoid and the myeloid. The myeloid are the large part, the dendritic are the tiny part. And the leukemia affecting this tiny part is extremely rare. And based on the blood of the patients affected by this leukemia, he developed a cell line, and a master cell bank, GMP approved by the French authorities, that we are still using today as a vaccinal platform. So the cell line have 3 key advantages. It's by definition, it's a cell line, so it's very easy to expand from a bag of the master cell bank. I will show it later. We can produce a lot of batches of cells. It's from the origins of the cells, and the plasmacytoid origins provide extremely interesting characteristic in terms of potency, and I will show you data. And last but not least, the first clinical trials feasibility done in melanoma in 9 patients shown that the cell line infused 3 times was not rejected. So it was possible to boost the immune systems by multiple injections. If you click again, you will see the last point. We are today in clinical stage. We're in phase 1-2 in non-small cell lung cancer, and the clinical trial has been initiated beginning of this year. Next slide, please. Yes, next slide. Yeah, if you press again. please, could you press? Yes, exactly. So what you see here is the pipeline of the company. Today we have three key elements. We are working for the future business plan, and we are today working on the business plan. But what you see here is the situation as it is today. For the PDC male, the melanoma clinical trials, the phase 1b has been completed, and the publication has been released a few weeks ago. We are today focusing on PDC lung, on non-small cell lung cancer, where, as I said, in phase 1/2 of clinical trials in France and Belgium. And we have in preclinical stage a neoantigens vaccine, which is a different kind of modalities where we want to use personalized antigen from a patient tumor. Next slide, please. What is important as well to mention, it's we have a strong link now with South Korea. We signed early 2019 a licensing agreement with LG Chem to develop the PDC lung cancer vaccine in Asia. So we granted exclusive license in South Korea and options in other Asian countries for the development and commercialization of PDC lung for lung cancer. So the plan is that they develop as well in South Korea and Asia the product in clinical trials, then they would commercialize it. So the total deal value is $123 million plus tied royalties on nail cell in Asia. Next slide. What you see after is the management team. So we have a quite strong management team with track record. Myself, I've been involved in several biotech companies. I've raised more than 130 million euros of funding. I've been involved in IPO, M&A, and Laurent Lévy, that you see here, is the co-founder of the company, CFO and co-founder, and Joël Plumat is the scientific founder of the company. He joined full-time from the lab in Grenoble in France, the company. And we have as well Claude Dédry, the VP of manufacturing, that has a long experience from GSK vaccine. And finally, the last arrival, Shanna De Bruyne, our medical director, that come from GSK vaccine and that led all the phase 3 clinical trials of the cancer vaccine of GSK, the MAGE study. So we have a very strong and experienced team with a track record. Next slide. Yes, what you see here is the process. So as I said, the cell line are based in a cell bank based in France, and we take a bag of the cell bank, and then we seed a single-use bioreactor. Can you click, please, on the slide? Can you click and you will, because there is an animations. Yes, so you see here the single-use bioreactors, and the cells can grow in suspensions in a very synthetic medium. And please click again. And after 9 to 10 days of cultures, we have billions and billions of cells that are produced. We just have to load the cells with peptide representative of the antigens, then irradiate the cells to stop the proliferations. Please, could you click again? Then we put, yes, then we put the cell in a off-the-shelf product ready to use from aseptic technology, and the cells are ready to be infused to the human to make the job, which is to amplify the CD8 T cells, cytotoxic CD8 T cells, to migrate to the tumor microenvironment to kill the tumor. Could you click, please, again? Yes, is what you see here in these illustrations. That's the full process of our vaccine today. Next slide. So I said the potency of our vaccine was very good, and this is an example of a benchmark study where we assess the potency in terms of capacity to expand cytotoxic CD8 T cells specific of one specific antigen, which is melanin. and we compare what you see in green with autologous and conventional dendritic cells, or in blue, the PDC line. And as you can see, the fold increase with our cell line is a lot more bigger, 20 to 200 for more potent than the classical approach. Next slide, please. In terms of IP, we have a quite solid positions. We have, of course, this unique master cell bank. We are the only one to get access to the cell bank, from exclusive access to the French blood bank. And we have as well 3 patents. 2 patents has been granted. The last one is in PCT, on the genetic optimization of PDC line. And at the top of that, we have, of course, all the know-how and data on the manufacturing, immunomonitoring assays, and all the preclinical data. Could you click? Yeah, click again. Yes, next slide. Yes, we did study on neoantigens. And neoantigens is a very hot topic currently. There is a part of the scientific community that is thinking that the immunity and of antigen specific of the tumor would be bigger and stronger, and this antigen specifically presented in the tumor and nowhere else would be strongly recognized by the immune system and would have a better effect in terms of activating the immune system. So to demonstrate if our cell line would work, or vaccine, or would work with neoantigens, we have selected 2 neoepitope, neoantigens, published in the literature, is what you see on the top of the slide, and where the mutations of the peptide in red has been clearly established. This peptide has been identified in the blood of patients or non-small cell lung cancer patients. And then we have loaded with this neoepitope the cell line and put in contact with the PBMC and the white blood and the CD8 T cells of naive donor, donor that have not been affected by any disease, on 3 different donors. And what you see on the bottom is the fold increase, after 20 days of culture with the no epitope, we have more than 150 fold increase thanks to the vaccine. So it shows clearly that the vaccine works extremely well in neoantigens in our case. Next slide. So the cell line is based on the blood of Caucasian patients. So it's the cell line is HLA-A2, and we have a minority HLA like B7 and B44. But we have already engineered the cell line. If you can click, please, by retroviral transductions, we have been able to enlarge the patient populations, and we know it works extremely well with the A24 or B45. A24 HLA is extremely common in Asia, for example. Could you click again? We have done, the cell line is really a platform. Today we are loading with GMP peptide that fixed to the HLA on the membrane of the cell line, but please click. We have as well done work on retroviral transductions, and if you click again, you will see that we have done as well transfection through tumor mRNA and through electroporations. Could you click? So you see we can really use the cell line with different modality according to the needs and the time constraints we have. Next slide. As I said earlier, we have done a study in melanoma, a first in human feasibility study, a phase 1B, which is a academic study on 9 patients where we have loaded a PDC line with 4 antigens, tyrosinase, GP100, melanase, and MAGE3. Could you click? And the result was quite interesting. We have seen that the tolerability was and safety profile was completely acceptable. That was the primary objective of this study. It was the first time we were infusing these cells to human. It's in this study in the 9 patients, we never observed any allogenic rejections. And last but not least, we have seen that the significant antigen-specific CD8 T-cell response in the blood of the patients detectable directly without any expansion. So that was extremely interesting in terms of demonstrating the potency of the cell line in human in vivo. Could you click again? And that this activity was interesting despite the fact we have only infused 3 times, and you will see that we believe we need to infuse at least 6 times to have a good clinical response. And despite as well, we didn't infuse co-administrate immune checkpoint inhibitors during this trial. So it was without any of these checkpoint inhibitors. Next slide. So checkpoint inhibitors, as you know, is a very hot topic. It has been a very provided a complete revolution in the treatment of cancer. The level of response for some patients is extremely good with the checkpoint, but there is still an unmet need on the non-responder. And what you see here is what we try to do to demonstrate the synergetic effect between our vaccine and the checkpoint inhibitor. And we have taken the blood of melanoma patients, and in melanoma patients, which is a very hot tumor, the immunosuppression induced by the tumor is extremely high, and we put in contact with the cell line loaded with one antigen, which is melanate. What you see at baseline PDC-VAC, we can, after 1 week of culture, have a 15-fold increase. If we use the anti-PD-1, which is the checkpoint inhibitor, we have almost nothing. It's normal because it's not this mechanism of action which is acting. But if we go to next slide, if we mixed both, next slide, please, we see this strong synergetic effect. No, the slide before. Slide before. We see that we pass from 15 to 35, so showing that definitely we have a synergetic effect where we co-administrate, where if we have an add-on of checkpoint inhibitor with the vaccine. It's why in the lung cancer, we are going to use for some cohort of the clinical trials, co-administration with checkpoint inhibitor. Next slide, please. Yes, what you see here is the design of the current clinical trial in non-small cell lung cancer, where the primary objective is a phase 1/2. Primary objective is again safety and tolerability because we are using different antigens from the melanoma. And the secondary objective is immunological activity and preliminary activity in B2 cohort. I will show you with and without checkpoint inhibitors. So we will test 2 doses of vaccine, low dose and high dose, and we'll start the 2 first cohort of patients in monotherapy, so without checkpoint inhibitor, and in adjuvant setting patients, so just after surgery and sometime chemical radiotherapy, a cycle of chemotherapy. And we have monotherapy low dose 6 patients. Then according to the safety, we'll go to monotherapy adjuvant, but 10 patients with the high dose, and we will go to the B1, what you see on the bottom right, in checkpoint inhibitor associations with the vaccine, low dose in 6 patients. Then we'll go to the B2, what you see on the bottom right, where we have 40 patients this time with high dose and vaccine, and it's in this specific cohort we hope to observe some preliminary activity of the vaccine. So as I said, we will infuse 6 times, and with this B1 and B2, it will be with checkpoint inhibitor. Next slide. To finalize my presentations, you see here the development plan and funding strategy. So we raised 20 million euros, as I said, end of last year, and with this money, we should be able to finance the full clinical trials on the 62 patients I've presented previously to measure safety, immune response, preclinical activity. We will carry on the work on the platform on the mechanism of actions of the cell line of the optimization of the cell line, working on neoantigens and development. And we hope in the range of the end of 2022 to launch a new fundraising for financing a phase 2 or phase 2/3 with clinical trials. With a new fundraising, and that we hope will be a randomized study. Next slide, please. So I would like to thank you for your attentions, and what you see here is part of the team when we have social events in Belgium. So I'm here for your questions now.
Okay, thank you very much, Eric, for your presentation. So have you ever participated in BioKorea before? Now, since you are based in Belgium, right?
Yes. No,
So-
I
oh,
didn't.
so-
It's my first participation to the BioKorea.
Oh, okay. So your first time, the kind of online as an online event, because this program is, undertaking every single year in Korea. But it's first time, of course, a new form as an online event. So, do you have many Korean partners, or are you looking for Korean partners?
Yeah, we have already Korean partners, as I told you, with LG Chem, which is our main partner. We have Korean investors with Korea Investment KIP, but we have as well 2 others that joined the round of investment. But we are still open to new partnerships for new indications of the platform, and to develop maybe a complementary technology to PDC line.
Okay. So, what do you think about bio industry in Korea? Because we have many potential investors and very good, investors. So, do you have any specific plan for the Korean market?
Yeah, Korea is a very interesting market because it's in a growing phase now. Yeah, and we have the government and has defined pharma, biotech, life science in general as a key focus, and we see the willing to develop biotech industries in South Korea, so which is extremely interesting. So our plan is definitely to carry on to reinforce the collaboration, of course, with LG Chem and to get maybe additional investor from South Korea and maybe over partnerships if we can in the future.
Okay, thank you very much. So I think we have to say goodbye this time around. Thank you very much, Ellie, for your wonderful presentation and also good, comments on Korean
Thank you.
bio
Thank you
industry.
very much.
Thank you.
Bye.
Bye. Thank you. Okay, ladies and gentlemen. So now you are with us a Bio Korea 2020 live talk. So this event actually has been prepared to introduce pharmaceutical, bio, and healthcare companies' core technology, as well as to share their strategies and visions to potential investors and partners. And now you're here with us at Live Talk. As a Live Talk is attended 28 companies to provide opportunity to find innovative technology products through real-time online streaming, right? So once again, thank you very much for your participation. We have about 100 viewers still with us, right? Thank you. And today, total of 8 companies are scheduled. Now you listen to 7 companies' introduction and their core technology and their vision and their pipeline. So I hope that it was very meaningful time to all of our listeners. And finally, we have one last company. And before that, if you are interested, please register our business forum at BioKorea 2020, and then you'll have a chance to connect the companies you are interested in. They're companies with core technology and high potential. So it's not too late. So please visit BioKorea 2020 Business Forum. Okay. So I hope that this event will provide a very valuable opportunity to strengthen cooperation between countries, not only in Korea. So actually, our program has started about 2 o'clock. 1.5 hours. Oh, okay. 2 o'clock. So it's been 1.5 hours, right? So we have one last company left. Before that, we're going to take a short break, okay? So maybe in 15 minutes, 13 minutes, we're going to meet our last company. Okay, so before that, we prepare a kind of promotion video. So let's watch the video clip. Thank you. Okay, so viewers, welcome back to BioKorea 2020 Live Talk, and thanks for tuning in almost till the end. So we have one last company left. Because this is the last presentation for today, we look forward to your comment, question, or anything, anything about your thought. So please leave your, comments on our live chat. So as I mentioned before, we'll give you a gift for our active participants. So we'll select some, participants and let you know in the last day in our official website. Okay? So the last company we're going to introduce is Thermo Fisher Scientific, one of the industry-leading pharma services solution for drug development, clinical trial, logistics, and commercial. Even with a short introduction, I'm very looking forward to seeing the presentation. So now it's time to meet our last speaker, Mr. Austin Shin and Pathion by Thermo Fisher Scientific. So, Mr. Shin, are you there? Hi.
Hi.
Hi, can you hear me? Okay, it seems to be okay. So, Thermo Fisher Scientific is one of the industry-leading pharma services solutions for drug development, right? So, okay, I cannot really hear. So can you just turn on the button, camera button on the screen? On the top of the screen. Turn on, please.
예. 지금 켰는데 혹시 안 나오나요?
Well, I turned it on, but okay.
You're here. Welcome. Mr. Shin, please start your presentation.
예. 프레젠테이션 시작하겠습니다.
We'll now begin the presentation. Good afternoon. I am Austin Shin from the Thermo Fisher Scientific. And it is my honor to introduce my company to all of you. And I would like to briefly talk about the company. We provide a lot of services and with different service lines. So I would like to give you the overview, and I would like to emphasize the quick to clinic in order to provide the drug substance DS and DP. And I would like to play a short video clip to explain that, and then we are going to have the Q&A sessions after watching the video. So I will now begin the presentation. And so again, it is my honor to talk about my company, and I would like to thank you, all of you, for giving me this opportunity. There is a company, Pathion, and another company, Thermo Fisher. And on the October of 2017, the Thermo Fisher acquired the Pathion. So we provide pharmaceutical services within the Thermo Fisher Scientific. And I believe that most of you are familiar with the Thermo Fisher. It is a global pharma company. About 70,000 people are working for this company. We are providing industry-wide services. And we have different sub-brands. Invitrogen, Invitramap, and Apla Biosystems, and their sub-brands. And we have the CDMO services under the Pathion. We provide the clinical manufacturing and productions, and we also provide the clinical labels and bioservices under the brand name of the Pathion. And we provide the labeling as well. Last year, we acquired the gene therapy specialized company last year. So we have a new brand as well. Next. We have the 6 business capabilities. First, ideas. We have chemical for the small molecules, and we have the API groups. And you're going to watch the video on the animals-based experiments and the development. and service lines. And this is the DS line. And for the DP line, we have the oral solid dose and the sterile fill finish. So we have these two big categories for the DP. And for the DP, we have different business units for North America and Europe. And I would like to talk about that later in more details. In addition to that, we have the soft gel products, a separate business unit. For the soft gel products, the needs are quite unique. So this is the reason why we have different R&D and manufacturing sites in different countries. And for the clinical divisions, we have the clinical trials from the Thermo Fisher Scientific, and we provide the packaging and labeling. Also, we all provide logistics, and we have depot services. We have one in Gimpo and Panghwa. And lastly, as I told you before, we acquired the new gene therapy specialized company, which has a specialty in viral vector services. So it provides or develops or manufactures gene specialized products. Next slide. And for Pathian, we are dealing with whole process from drug delivery to the logistics. And after it is acquired by Thermo Fisher Scientific, many lines were added in order to satisfy the needs of the customers within the time restraints. That is the biggest strength. And we already manufacture more than 20 billion solid doses and 156 million sterile doses. And also we provide the support for the development for the small molecules, more than 200 for the large molecules, more than 50. We dealt with the technology transfer and manufacture and delivery for the small and large molecules, and we are working with more than 900 customers. And as you can see from this slide, we provide the services for the whole life cycle from generic. So we have integrated operation system for the whole process. Please, next slide. We have about 55 different sites across the globe. There are biologic-related sites, viral factor-related sites, and we have sites for the clinical labeling and packaging. Most of the sites, as you can see from this side, are in North America or Europe. In particular, the one in red means the sites for the clinical trial supply. And these are the depot for the clinical services. We have all these sites across the globe, and each site is dealing with the development and manufacturing. And it is also dealing with the main regulators such as FDA and EMA, and it is working in coordinates with the regulation of the FDA and EMA. Next slide. As I previously explained, we have a large spectrum. We start from the new drug development, and we are dealing with the commercial packaging, and we provide services for the whole life cycle of the drug development. And for all services, we have separate and bundling services. So we can provide multiple CDMOs and partners who can involve. However, we select one CDMO to deal with the whole process or the whole life cycle. This has advantage. One of them is easy technology transfer, and we can save some unnecessary expenses and time. And also, we are dealing with from preclinical services to the last stage of the clinical test. And if you like to move on from the preclinical stage to the another level of the stage, you don't have to change our CDMO. We can flexibly respond to that because we are dealing with the whole process, whole cycles of the clinical trials. Can you move on to the next slide? And we have, in terms of the number of the NDAs for the 10 years, we got 107 NDA approvals from 2008 to 2018. When you compare to other outsourced approvals, it accounts for 19%. In terms of the number of the NDAs, or in terms of support for top performing drug products, we are outperforming compared to other competitors. This proves we are number one in CDMO in the area, and the survey proves that we are providing excellent and world's best services. Please move on to the next slide. This is also what I explained before. We are cooperating with the global companies in the area of biopharmacy, biotechnology, and we are cooperating with 17 of the top 20 specialty pharmaceutical companies. We have more than 900 clients. 80% of them have new compounds. And also we can now provide the vital factor services customized to the customer needs. Next slide. Thank you for your time. And we have the virtual booth under the name of Thermo Fisher. So if you have any questions regarding Thermo Fisher or Pathian, please feel free to contact me with an email, or if you have time, please visit our virtual exhibition center. After that, as I told you before, we are going to watch short video, and then we are going to have the Q&A sessions. Thank you. Please play the video.
네, 저희 영상으로 플레이하도록 하겠습니다.
Hi, I'm Frank Ricacco from Thermo Fisher Scientific, and I'm happy to talk to you today about some new approaches and technologies that we are using to enable accelerated development and manufacturing timelines for biopharmaceuticals. Thermo Fisher Scientific is a leading global biologics CDMO, supporting our customers with drug substance and drug product manufacturing facilities at locations around the world. As a company, we have over 30 years of GMP manufacturing experience. We focus on mammalian expression systems. Our capabilities include cell line and process development for early and late-stage molecules, and clinical and commercial manufacturing with over 32,000 liters of single-use GMP capacity currently. As you can see from this slide, we have extensive experience with tech transfers and development programs for multiple molecule types, and we can offer integrated gene-to-patient solutions, including drug substance, drug product, and Fisher Clinical Services to provide all of your manufacturing and logistical needs under one roof. The past few years have seen a dramatic growth in the number of approved biologic therapies, and each year, more and more biologics are given a fast-track designation or breakthrough therapy status. As a result, biopharmaceutical companies are looking for approaches and solutions to enable accelerated development timelines and a faster path to the clinic for their breakthrough molecules. There are a number of critical items to consider when trying to reduce development and clinical manufacturing timelines. The goal is to balance speed, risk, and robustness, understanding where you can take appropriate risks in development, while still generating the quantity of material needed to enable your clinical trial and without sacrificing product quality. In order to address these needs, Thermo Fisher recently launched the Patheon Quick-to-Clinic offering. With Quick-to-Clinic, we can save 7 months compared to a traditional program, providing released GMP drug substance in just under 14 months, or released drug product in 15 months. Quick-to-Clinic leverages our deep experience in biologics, combined with our in-house platform processes and raw materials to provide our fastest timelines to GMP drug substance or drug product release, specifically for IgG1 and IgG4 monoclonal antibody molecules. These development timelines are enabled by the use of well-established platform processes for monoclonal antibodies, high-throughput automation technologies such as the Beacon, Amber, and TCAN systems in our development labs, multiple development activities conducted in parallel, and standardization of raw materials and documentation to streamline development and manufacturing while minimizing risk to product quality and regulatory filing. The Thermo Fisher platform for biologics development and manufacturing is flexible. We follow a very familiar process commonly used for monoclonal antibodies, similar to those used widely across the industry. We leverage Thermo Fisher cell lines, raw materials, and equipment wherever possible to streamline procurement and development activities, but we can also accommodate materials and equipment sourced from other vendors within our platform. For example, we can use Thermo Fisher single-use bioreactors, but we also have other manufacturers' bioreactors, such as GE Accelerex, within our platform. A quick-to-clinic program will deliver a cell line, a process, tox material, all necessary reports, and approximately 1 kilogram of released GMP drug substance for your clinical trial in 14 months, from DNA to drug substance. 15 months includes drug product manufacturing. This slide illustrates at a high level the quick-to-clinic timeline, with cell line development taking about 4 months, delivery of non-GMP tox drug substance at month 8, GMP drug substance at just under 14 months, and drug product at month 15. This slide shows a bit more detail of the overlapping activities taking place during the process development and manufacturing timeline. Cell line generation is performed using a Thermo Fisher cell line, shown here as Freedom Cho S. Automated systems such as the Beacon, Amber 15, and Amber 250, and the TCAN Freedom Evo are used to streamline process development. After confirmation of the process at the bench scale, a 200 or 250 liter pilot run is conducted to generate material for toxicology and viral clearance studies, and to provide confidence in scale-up of the process to 500 or 2,000 liters for GMP manufacturing. Our platform has, until now, utilized the Freedom Cho S cell line. But later this year, we will be launching Freedom XB Cho S, a new, higher titer, stable cell line system for expression of biologic molecules in CHO cells. This cell line is based on the very popular XB Cho transient expression system, but has been adapted for stable expression and high titers with an all-new vector system. The system is designed to provide antibody titers in the 3 to 5 gram per liter range within a CLD timeline of 4 to 5 months and without royalty or milestone payments, just like the existing Freedom Cho kit. Here's some data showing how Freedom XB Cho performs. The bar graph on the left shows a comparison of monoclonal antibody titers in XB Cho S compared to an HEK 293 cell line and an older Cho S cell line. The differences are quite dramatic. The line graph shows titer data from 3 clones taken from a single XB Cho S transfection, with monoclonal antibody titers ranging from 2.5 to 3 grams per liter in this instance. Further, we have observed consistent product quality between transient and stable expression of a molecule in the XB Cho system, allowing our customers to confidently move from transient expression at the R&D stage to stable expression for development of a manufacturing process with low risk of significant changes in product quality. And lastly, I want to highlight that the Freedom XB-CHO kit will feature a new 2-vector system, which will allow for good control and tuning of heavy and light chain ratios to optimize antibody expression. Here's some scale-up data of a Freedom XB-CHO S cell line using Gibco's stable production medium optimized for it, called XB-CHO SPM, showing good growth and antibody production in a Thermo Fisher single-use bioreactor without any process optimization or loss in titer compared to the bench scale. And for our cell line development programs, including Quick to Clinic, we are now utilizing the Berkeley Lights Beacon platform for cell line development. This is a very exciting technology, which we were one of the first CDMOs to offer to customers. The Beacon performs single-cell cloning on a chip, using light to move single individual cells into pens holding only 1 nanoliter of media. And the system can visualize growth and antibody production at the nanoliter scale. This instrument gives us the ability to evaluate 7,000 individual clones at once with assurance of monoclonality, allowing for faster cell line development timelines and the screening power needed to identify the highest producers in a population of transfected cells. I hope you've enjoyed this presentation. Please feel free to reach out to the experts at Thermo Fisher Scientific if you would like to learn more about our capabilities as a leading global biologics CDMO. Thanks for your time.
Okay, so we just had an English version of your presentation, right? Mr. Shin, are you there still?
Oh, yeah.
Okay. Okay. So we got some comments. Also, I have a question to your company. So you mentioned 가상 전시를 참여하고.
You mentioned about the virtual exhibition. Can you see my face? I'm going to soon appear.
Be soon on the screen. Well, I believe that you can hear me well, right? Okay. 자, 나왔습니다. 잠시만.
Oh, actually, can you give me a second to adjust it? I think you're participating in the virtual exhibition. So, in addition to the presentation, can you talk about what additional information that people can get if they visit the virtual exhibition? Yes, we have the virtual expansion specialized for each cell line or each service line. So I believe that you can get some details because today I just briefly explained about the service lines at the company. So people can get the more detailed information, and I think there is a direct way to contact you, right?
This forum of Bio Korea 2020 for more details and information. 자, 그리고 지금 저희 투자자들도 있으실 수.
I think many investors and many people are interested in the company.
마지막으로 또 하고 싶은 말씀이 있으실까요?
We have a lot of Korean customers. And people are interested in the new drug development and manufacturing. And we have a lot of service lines. So if you're interested in one of the service lines, please visit our homepage. And we have a YouTube channel. So please visit our YouTube channel as well in order to respond in a swift manner. Thank you. Thank you. So it was the very last presentation.
Thank you very much. 이제 보내드리도록 하겠습니다. 고맙습니다. Thank you. Okay, so Thermo Fisher Scientific, one of the industry-leading pharma services solutions for drug development, clinical trial, logistics, and commercial manufacturing. That was the last company for today. So, viewers, ladies and gentlemen, how did you like live talk today? So it's kind of a new try. It's a very new attempt. So it was not perfect because it's today, first day. But I think we can find various possibilities in this era of social distancing. Once again, thank you very much for your active participation. And this live talk will continue until Friday. So we have today is Wednesday. So we have 2 more days. So please don't miss it. Stay with us until Friday. So again, on the last day, we'll select some participants who participated very actively on our live chat. We'll give you some gifts. Okay? So thank you very much. And I have to say goodbye for today. I'll meet you tomorrow at the same place and the same channel at 1 p.m. Thank you. Bye-bye.